Good day, everyone. I hope you're all doing okay. Before we proceed with our lesson about meiosis, let's have a quick recall through this short video. So, this is a cell undergoing mitosis. Now, I want you to watch this video carefully and then try to identify each mitotic stages correctly. Okay. Let's pause the video. What's mitotic stage do you think is this? Good job, it's prophase. Now, what mitotic stage do you think this is? Good job, it's metaphase. Now, what stage or what mitotic stage do you think this is? Very good, it's anaphase. Now, what stage is this? Good job, it's telophase. And then lastly, we have cytokinesis or cytoplasmic division. Congratulations if you were able to identify each mitotic stages correctly. So now we'll proceed with the next type of cell division, which is meiosis. If you want to read more about meiosis or the cellular basis of sexual reproduction, then you may refer to our Cengage book, and it is at chapter 11. So here are our learning objectives. If you can notice, these are just the same learning objectives that I've discussed or I've read to you before in mitosis. But for our lesson, of course, we'll be focusing on the different stages of meiosis, and then we will compare and contrast mitosis and meiosis. So we will discuss the difference between these two cell divisions. Let's start. Okay, so this is a scanning electron image of an egg cell and a sperm cell. And if you can notice, the egg cell is comparably larger or bigger than the sperm cell. So this is actually their actual size. And these egg cells and sperm cells, they are necessary for what kind of reproduction? Very good. Very good. Sexual reproduction, which is defined as the production of offspring through the union of male and female gametes. So gametes, this is our general term for sex cells, or for example, egg cell and sperm cell. And meiosis matters because it is a specialized process of cell division which produces these gametes or sex cells. So without meiosis, then sexual reproduction is not possible. Okay, now gametes have half the number of chromosomes present in the body cells of a species. So for example, if humans have 46 chromosomes in their body cells, that means how many chromosomes are there in their gametes? It is very good. It is 23 because 46 divided by 2, or the half of 46, is 23. So the egg or the egg cell contains 23 chromosomes, and then the sperm cell also contains 23 chromosomes. And when they are united together, or when they undergo fertilization, the sperm and the egg cell, it will produce a zygote with 46 chromosomes. So this means that from becoming, through the union of two haploid sex cells, then we get, or it is, the result is a diploid cell. Now, have you ever heard of the phrase everyone is unique? Well, meiosis takes that seriously because the possibility or the probability that two children of the same parents could receive the same combination of chromosomes is one chance out of 70 trillion. So that's a very, very, very small probability. So, in other words, yung chance na sobrang kamukha mo or sobrang kasame mo ng genes yung kapatid mo is 1 in 70 trillion because of different events that happen during meiosis, such as recombination from crossing over. So later on, we will discuss kung ano ba yung crossing over and how it contributes to the genetic variation or genetic variability. So yung only exception for this small probability is yung identical twins which arise from mitotic division of a single fertilized egg. Or yung sinasabi lang nito is that yung fertilized egg na isa ay nahati sa dalawa. Kaya nagkakaroon tayo ng identical twins. Okay. So, in humans and other animals, meiosis takes place in the primary reproductive organs, which we call the gonads. For males, yung mature gonads, we call it the testes. And it produces the spermatozoa, or what we commonly know as the sperm cell. On the other hand, for females, the mature gonads are called ovaries, and it produces the ova or the egg cell. And then, syempre, before maging egg cell and sperm cell, itong mga gametes natin, first, they were a germ cell. Okay? Germ cell muna yung tawag nila sa kanila. Sorry. These germ cells will undergo what we call gametogenesis, which is the process of gamete formation. So again, these germ cells will undergo gametogenesis to become what we know as the sperm cell and egg cell. So gametogenesis, if hihimayin natin yung root words niya, from the word genesis, which means start or beginning, and gamete or gameto, which means gamete, so start or beginning of gametes. Okay. Now, I have here a diagram of meiotic division or meiosis. So, if you can notice, it is very different from mitosis. So, can you spot all the differences? You may pause this video and then write down kung ano ba yung na-notice nyong differences sa notebook nyo. Okay, so yung first difference ng meiosis sa mitosis is that meiosis has two cell divisions. We have the first meiotic division and then the second meiotic division. The first meiotic division produces two daughter cells. On the other hand, the second meiotic division produces four non-identical daughter cells. Because if you can recall, in mitosis, yung naproproduce na daughter cells, na two daughter cells, they are identical to each other, right? But in the case of meiosis, the daughter cells, or the chromosomes that the daughter cells carry, they are not identical. Aside from the having two cell divisions, meiosis also has this event which we call crossing over. So later, madidiscuss natin kung ano ba yung nangyayari during crossing over. And then, aside from that, sa meiosis, nababago din yung chromosome number natin. Na-discuss natin again na yung gametes natin, it has half the number of chromosomes a somatic or a body cell has. So, dito, pagdating or after second meiotic division, yung number of chromosomes natin ay kalahati na per cell. Unlike sa mitosis na napreserve or equal pa rin yung chromosome number ng daughter cell sa parent cell. Okay? Again, for meiosis, yung number of chromosomes for daughter cell is already half the number of the chromosome of the parent cell. On the other hand, for mitosis, the number of chromosomes for the daughter cell is still the same as the parent cell. Okay? Okay, so let's proceed. Now, before we learn about the different stages of meiosis, it is imperative that we learn about alleles. So what are alleles? According to these, the alleles are the versions of each gene present in the members of a homologous pair that may be the same or different. So what this is just saying is that alleles are the alternate versions of a gene. So for example, we have two homologous pair of chromosomes here, or a pair of homologous chromosomes. When we say homologous, that means they are similar. Okay, so here we have an allele for the color of the flower. So sa left side, yung color ng flower or the allele for this flower codes for white. On the other hand, yung allele for the right chromosome codes for purple or codes for the color purple. So you see, even though they have or yung allele nila is located sa same position ng chromosome or matatagpuan sila sa same region ng chromosome, they are different kasi magkaiba yung characteristic or yung kinocode nila for color. Yung isa, it codes for white color. Yung isa naman, it codes for purple color. So, if sabihin natin na yung allele for so, let's say, if yung allele natin for purple flowers is the dominant allele, and then yung allele for white flowers is the recessive allele, then pinag-combine natin sila. What do you think will be the resulting color of the flower or the progeny? Progeny is yung anak. It will be purple, right? Unless there are different or there are exceptions. Since meron din tayong tinatawag na different allelic or gene interactions, which is yun yung mga codominance, mga ganun. But that's not our focus for today. So, ayan. Yun lang muna for now. Sneak peek. Ayan. So, each individual, except again for identical siblings since they carry the same genes, has a unique combination of alleles in each homologous pair. So now, let's proceed with the different stages of meiosis. So we will first start with the first meiotic division, or what we call meiosis 1. So meiosis 1 has the 4 stages then sa cell division, which is the prophase, metaphase, anaphase, and telophase. But for meiosis or meiosis 1, maglalagay tayo nung 1 to indicate na yun yung first meiotic division. So, instead of calling it prophase, we call it prophase 1. Okay? So, this is what happened during prophase 1. If you can notice, meron tayo ditong tinatawag na tetrad. Here. Tetrad. So, tetrad, kaya natin siya tinawag na tetrad because it consists of 4 sister chromatids. Again, it is called a tetrad from the word tetra, which means 4, because it has 4 sister chromatids paired together. So, another term for tetrad is bivalent. Kung ayaw nyong tandaan ng 4 sister chromatids na tetra, just think of bivalent. So, when we say bi, di ba, dalawa? And then, bivalent means 2 chromosomes paired together. Yan. So, yung tetrad natin, it will undergo crossover or crossing over. Ma'am, kanina pa natin, or kanina mo pa binabanggit yung crossover. What is that? Well, Ito na yung crossover or crossing over. So again, yung sister chromatids natin or two homologous chromosomes, they will align and then pair with each other. So, kapag nag-pair na yung homologous chromosomes natin, they will become a tetrad or a bivalent. This process is called synapsis or synapsis. Again, when two homologous chromosomes undergo pairing or pair with each other, they form tetrads or bivalents, which we call synapsis, or this event is called synapsis or synapsis. So after synapsis or after the pairing of homologous chromosomes, these homologous chromosomes will physically exchange segments, which is the crossing over. So basically, during crossing over, nag-e-exchange gift lang yung chromosomes natin or nagpapalit lang sila ng segments. Kaya kita nyo, ayan, para silang nag-holding hands na pa-cross. And then, after crossing over, yung pinagpalit nila na segments, it will be marked by thickened spots, which we call the chiasmata. Again, yung segments na in-exchange ng dalawang chromosomes, they are called the chiasmata, and they are marked by thickened spots under the microscope. Okay, so again, review natin kung ano yung nangyari sa prophase 1. Prophase 1 pa lang to ha. First, yung homologous chromosomes, they will align and then pair with each other, forming tetrads or bivalents. And this event is called synapsis. After synapsis, these homologous chromosomes will undergo crossover or crossing over. During crossover, the sister chromatids of these homologous chromosomes will physically exchange segments. And these segments will be marked by thickened spots, which we call chiasmata. Okay? Yan. So, prophase 1 pa lang, marami nang nangyayari sa meiosis. That's why sobrang different niya sa mitosis. Kasi 'di ba, if you can recall, sa prophase ng mitosis, ang nangyayari pa lang is yung condensation ng chromatin into chromosomes. And then yung disappearance ng nuclear envelope natin to let out the chromosomes into the cytoplasm where cell division will happen. So after prophase 1, what is the next stage? Okay, very good. So it is metaphase 1. So here, another visualization lang din ng crossing over or recombination by crossing over. So ayan, we have again our homologous pair, and then they will undergo crossing over or crossover. After nila mag-physically exchange ng segment, pero na silang chiasmata, and then they will undergo cell division, producing 4 non-identical daughter cells. Ayan, so again, after prophase 1, we have metaphase 1. So just like in mitosis, sa metaphase 1, mag-a-align lang din yung chromosomes natin at the metaphase plate. But now, here's the difference, and you need to take note of this key difference. Instead of univalents or single chromosomes, ang mag-a-align na sa metaphase plate during metaphase 1 is our bivalents or our tetrads. Yan. So you see, meron tayo ditong dalawang chromosome. We have the blue chromosome and then the red chromosome. So that is its difference from metaphase of mitosis. Kasi again, sa metaphase ng mitosis, ang mag-a-align lang doon is the univalent or the single chromosome. On the other hand, for meiosis 1, specifically metaphase 1, ang mag-a-align sa metaphase plate is our bivalent or our tetrad. Okay, next, after metaphase 1, of course, we have anaphase 1. So, during anaphase 1, instead of sister chromatids, ang maghihiwalay or ang mapupul towards opposite poles is yung tetrads or yung homologous chromosomes nung tetrads. Again, instead of sister chromatids, ang mapupull towards opposite pole during anaphase 1 will be the homologous chromosomes. Okay? Then, after anaphase 1, of course, we have telophase 1. So, sa telophase 1, very similar siya sa telophase ng mitosis in which magdi-decondense lang yung chromosome natin into chromatin and then the nuclear envelope will start forming again para ma-enclose yung ating chromatin. So, mabubuo lang ulit yung nuclei or yung nucleus natin during telophase. And then, instead of cytokinesis, iba yung term natin. We call it interkinesis. Again, instead of calling it cytokinesis, we call it interkinesis. Because, ito yung cytokinesis that happens between the first meiotic division and second meiotic division. So, nasa gitna siya. Basically, same process as cytokinesis na iba lang yung pangalan niya kasi it happens between the first meiotic division and second meiotic division. So, after meiosis 1, we will proceed with, okay, you guessed it right, meiosis 2. So, this is what happens during meiosis 2. And it is very, very, very similar to mitosis. Kasi, during prophase 2, same process lang din siya during prophase of mitosis. So, the chromatins will condense into chromosomes. The early mitotic spindle fiber will start to form from the centrosome. And then, slowly mag-a-attach siya dun sa kinetochore that is located at the centromere of our chromosomes. And then, during metaphase 2, the chromosomes will align at the metaphase plate or the equatorial plane. Then, during anaphase 2, the sister chromatids of these chromosomes will be pulled towards opposite poles or will move towards the opposite poles of the cells. And then telophase 2, again, the chromosomes will decondense into chromatins, nuclear envelope will start to form, forming two daughter nuclei, and then it will be immediately followed by cytokinesis, in which it divides the cytoplasm or divides the cell completely, producing 4 daughter cells. So, if you can notice, kaya 4 daughter cells yung naproproduce dito. Kasi sa meiosis 1, di ba, we have 2 daughter cells. So, yung dalawang daughter cells na yon, they will both undergo meiosis. So, kung yung 2 hinati pa na, kung yung 1, yung isa ay hinati natin sa half, then that will produce 2, di ba? Tapos yung isa hinati ulit natin sa half, that will produce another 2 cells. So, pag pinag-add natin, 2 plus 2 is equal to 4 daughter cells. But, yung difference niya sa mitosis is that yung chromosome number per cell is half of the parent cell, and the chromosomes or the cells, they are non-identical to each other. Because again, nagkaroon tayo ng recombination by crossing over, kung saan nag-exchange sila ng segments. So, you see, yung meiosis 2, very similar lang talaga siya sa mitosis kasi wala na tayo nung tetrads. Yung univalence na lang ulit yung ina-align natin and pinapaghiwalay during metaphase and anaphase respectively. I hope that's clear. Okay, now I have a question here. You can again pause this video if you want to think. So, how does meiosis contribute to genetic variation? Yan, nasabi ko na or na-mention ko na to earlier that meiosis contributes to genetic variation. But how exactly? Well, meiosis contributes to genetic variation through a series of 3 events. So, yung una is the crossing over or the crossover that happens during prophase 1. So, 'di ba sa crossing over, nagpapalit or e-exchange ng segments yung pair of homologous chromosomes natin. So, dun pa lang, meron na agad variation. And then next, after the crossing over, is the independent assortment that happens during anaphase 1. So, sa independent assortment, this just means that yung paghihiwalay nung tetrads natin or nung bivalents natin, it is randomized. Meaning, we don't know which side mapupunta yung, for example, yung blue na chromosome, di natin alam kung mapupull siya towards the left or towards the right part of the cell. And yung, consequently, yung red chromosome natin, for example, di natin alam if mapupunta siya sa left or right. So, it is completely random. So, that's another source of variation or variability. And then, lastly, is the particular sets of female and male gametes that unite in fertilization. So, ayan. We don't know kung aling sets or aling sperm cell yung mananalo and aling egg cell yung ma-fertilize during fertilization. So, since yung egg cells natin and sperm cells natin, they carry different sets of chromosomes. Okay, so, para mas madali nyo maintindihan yung sources of variability, you can think of it as this analogy. So, for example, we have a deck of cards. And then, you shuffle those deck of cards. So, that means randomize na yung order ng cards natin, right? So, that's the first event, which is crossing over or crossover during prophase 1. And then, from those deck of cards, hinati mo sila into 4 sets. Again, from those deck of cards na shuffle mo, hinati mo or you divided it into 4 sets. So, that's the independent assortment. And then, yung 4 sets na yon, you randomly pick 2 from those 4 sets. Again, from those 4 sets, you randomly pick 2 sets. So, that's the third event, which is the fertilization or the union of male and female gametes. So, you see, completely randomized and shuffled yung deck of cards mo or yung genes mo. Kaya, we have sources of variability or genetic variation. So, we've been talking about genetic variation, but what exactly is the importance or the purpose of genetic variation? Again, you can pause this video to think about this question. Okay, so genetic variation is important because it is what helps us adapt sa changing environment natin, or it is what helps us evolve. If you want to take it to a much larger scale, it what helps us evolve and adapt to our changing environment. Because imagine if there are no changes dun sa traits natin, same lang siya generation after generation after generation, but yung environment natin, it keeps changing. So what do you think will happen if ganun yung case? Diba, there is a big possibility or big probability that we will go extinct. Kasi yung environment natin changing, but you yourself, you're not changing. So that's why genetic variation is important, so we can adapt to our changing environment. So now, let's compare the differences, or let's compare and contrast meiosis and mitosis. So, you can try answering this on your notebook or on a piece of paper. Just copy this table and then answer. So, for chromosome number of daughter cells, again, for mitosis, ilan kaya to? Okay, very good. So, for the chromosome number of daughter cells in mitosis, it is the same number as the parent cell. On the other hand, for meiosis, the chromosome number of daughter cells is half the number of the parent cell. So, for example, sa mitosis, meron tayong 46 chromosomes per cell. So, sa meiosis, if half na nung 46, then ang kalalabasan niya na chromosome number is 23 per cell. And from being a diploid, yung cell natin, it becomes haploid. Kasi again, isang set na lang or half the set of the diploid na lang yung meron tayo during meiosis. Now, for the number of cell divisions, they are also different. So, what do you think is the difference sa number ng cell divisions for mitosis and meiosis? Okay, good job. For mitosis, we only have one cell division. On the other hand, for meiosis, we have two cell divisions. The first, meiotic division, and then the second, meiotic division. Sa stages, yung difference nila, yan, masyadong marami, but we can just say, sa mitosis, we have the PMAT, prophase, metaphase, anaphase, telophase. And then for meiosis, we have PMAT1 and PMAT2, or P1, M1, A1, T1, P2, M2, A2, and T2. So, those are the stages. So, in total, meron tayong 4 stages only sa mitosis. On the other hand, for meiosis, we have a total of 8 stages. And then, the presence of synapses or synapsis, of course, it is absent in mitosis and it is present in meiosis. For the presence of crossing over, again, absent in mitosis but present in meiosis. So, both synapses and crossing over, they happen during what phase of meiosis? Very good. It happens during prophase 1. Now, for the cell type that undergoes division, for mitosis, it is what type of cell? Very good. It is somatic or body cells as well as germ cells. Okay, and then for meiosis, the cell type that undergoes division is only the germ cell. Okay? Again, for mitosis, both somatic and germ cells undergo mitosis. On the other hand, only germ cells undergo meiosis. Now, for the genetic consequences of mitosis, mitosis produces identical daughter cells. On the other hand, meiosis produces non-identical daughter cells. So, I hope that's clear. So, here's the complete answer. Ayan, pwede din kayong maglagay ng PM dito or M to symbolize or signify pro-metaphase. But usually, di na din natin ina-add yung pro-metaphase. It's just a transitional stage from prophase to metaphase. Okay, so here is another table summarizing the differences between mitosis and meiosis. Okay, so ayan. Nadagdag lang dito is yung role in life cycle. So, for meiosis, ayan nga, it is needed to produce gametes for sexual reproduction. And for plant cells, it produces spores, as well as for fungi and algae. And it generates genetic variability, or it is a source of genetic variation in gametes or spores. Okay, but for mitosis is usually used for growth, sexual reproduction in some eukaryotes and mostly in prokaryotes. Because if you can recall, prokaryotic cells or prokaryotes usually don't undergo or don't have sexual reproduction. What they have is asexual reproduction, just like binary fission, which is just essentially cell division. Okay. So, ayan. Ito, interesting fact lang. It's not really that essential na malaman nyo. So, interesting fact or trivia lang. So, this is entitled, Twin, Where Have You Been? Ayan. So, for example, si Isnir and si Dr. Jose Rizal. If you've been watching PBB, viewers and fans, they've been comparing Esnir and Dr. Jose Rizal kasi magkamukha daw sila or very similar yung facial features nila. So, how is mitosis and meiosis related to twins? Yeah, so we have here a question. How are identical twins formed? If you've been listening, ayan, na-mention ko earlier how identical twins are formed. So, for example, we have George and Jai. I don't know if kilala nyo pa sila, but they were also part of a PBB. So, naging housemates sila sa PBB, si George and Jai. And then, how are fraternal twins formed? So, yung example natin ng fraternal twins ay yung dalawang vocalist or dalawang singers natin sa Ben and Ben. Yan, they are actually fraternal twins. Yan, so here's our trivia. Identical twins come from one fertilized egg that splits into two embryos through mitosis. On the other hand, fraternal twins come from two separate eggs, each fertilized by a different sperm through meiosis. So you see, even dun sa twins natin, sa identical twins and fraternal twins, mitosis and meiosis are involved, or they play a role in the birth of these twins. So let's further discuss that. Based on this picture or this illustration, we can clearly see the difference between between how monozygotic twins or identical twins are produced versus how dizygotic or fraternal twins are produced. For monozygotic twins or identical twins, we only have one fertilized egg. This fertilized egg will split into two through mitosis. Again, this fertilized egg will undergo mitosis and then split into two. That is why 100% na same yung genes nung kapag identical twins, kasi they come from one fertilized egg. On the other hand, for fraternal twins, they come from one egg cell and then one sperm cell and then another egg cell and then another sperm cell, and then na-fertilize sila sa iisang womb. So we have one egg cell and one sperm cell, then another egg cell and then another sperm cell. And hindi possible yung fertilization or yung union nitong gametes natin, egg cell and sperm cell, without the help of which cell division? Very good. Without the help of meiosis, diba? So, kung wala tayong gametes, wala tayong fertilization or wala tayong sexual reproduction. Kaya, meiosis is important so we can produce gametes. So, going back, ayan, yung fertilized egg natin, each of the fertilized egg will undergo further cell division until maging zygote sila. That's why, just like sa siblings nyo, 50% lang din yung similarity ng genes for fraternal twins. Yan, kasi they come from different egg cell and sperm cell. Magkaiba sila ng egg cell and sperm cell. Kaya 50% lang. Unlike sa identical twins na nanggaling sila sa iisang egg cell. Kaya 100% same sila ng genes. Ayan. You might be asking, bakit sa egg cell ng fraternal twins, hindi siya nag-split into two just like sa identical twins? Well, yung reason for the splitting of one fertilized egg cell into two is still unknown, or it is still a mystery for scientists, geneticists, doctors, and alike. So, it's still a mystery and the cause is still unknown. That's why yung identical twins, they are a rare occurrence or can be considered a miracle. Yan. So that's it for our lesson about meiosis. I hope yung different stages of meiosis as well as its difference from mitosis was clear to you. If you have any questions, you may message me or you may ask me about them during our face-to-face sessions. Okay, so goodbye class, and thank you for reaching or for watching this video until the end. Bye bye. Thank you.